Okay, so this doesn't need much introduction. I mean, we all agree that the heart adapts to a biomechanical stress via a default pathway that lead to more fibrosis and a fewer myocytes, basically, and hypertrophy. But what we want is more myocyte and less fibrosis. So the whole idea is to find ways of inducing some kind of, I mean, reversing the process and trying to induce some kind of regeneration. So there's many ways you can imagine to, I mean, envisage to achieve this goal. But our laboratory is more interested in trying to find or identify pathway that might recruit, mobilize, maybe induce to differentiate endogenous cardiac stem cells. So there's basically two approaches. I mean, we can use a candidate pathway approach or more unbiased approach. And I will talk a little bit about both today. And I will start with this candidate pathway approach, as I call it. And we have a particular interest in the notch pathway in this context. So this also doesn't need much introduction. We've had it already. But so I just want to remind you that the notch pathway is just a communication system between two adjacent cell types. One cells sending a signal that express a ligand, and there's jagged 1, 2, and delta like 1, 3, and 4 as ligands. In the heart, the major ligand, the prominent ligand is jagged 1 in the adult heart. And the signal is sent to a signal receiving cell that express receptor, notch 1 to 4. And in the heart, the prominent form is notch 1. Now, we're obviously interested in the notch pathway because it has a role in cardiac development, but also in self-renewing capacity of many regenerating adult organs. And we have shown in the past that it has a crucial role in the normal adult heart. So this is just as an introduction. This is old data, but what is the role of notch in stem cells? So I just want to remind you that if you take the prototype stem cell, the embryonic stem cell, if you differentiated them here using the hanging drop models, basically you have a downregulation of the notch pathway. I mean, certain components of the notch pathway are downregulated, and signaling is turned off during differentiation. Now, in terms of cardiogenesis, we used notch 1 deleted cells, and what you observe in these cells, these cells have more capacity to produce cardiomyocyte, basically. So it's early on because they have more commitment to the mesodermal lineage, and more specification, and maybe more differentiation, and at the end you have maybe a more mature cardiomyocyte, and if you look at EBs, you have more beating EBs, more beating area per EBs. So notch is basically in these cells an inhibitor of cardiogenesis. Now this is also seen in just wild-type cell. If you just use this ES cell line that express GFP under control of the NKX2 promoter, and you just sort for notch 1 low cells, basically already expressed quite a large amount of brachyury, even in undifferentiated cell that has been maintained in LIF and MEF. And if you look at later time point in differentiation, basically you see that the notch 1 cells are the cells that express MES1 and obviously NKX2.5, the notch negative cells. Now, in postnatal cardiac stem cells or progenitor, I mean you can isolate this cell, for instance, from the neonatal heart, and use them to differentiate into cardiomyocyte in vitro. This is a very simple experiment. You can switch into a differentiation medium. At the beginning, you don't have much, but at the end, you have fully differentiated cells that even beat. Now, in these cells, I mean, this cardiac precursor cell can even be transferred in vivo, and we have used this model that we have established. It's a neonatal animal. We inject precursor into the temporal vein, and the cells then contribute to the normal growth of the heart after birth. Now, if you look at these cells and look for notch signaling in these cells, what you observe that when you maintain these cells in expansion, basically they do receive a notch signal, and they are a precursor because they express NKX2.5. But when you start to differentiate these cells, then these cells start to downregulate the notch signaling, basically. And at the end, you again obtain cardiomyocytes that essentially are a notch negative, notch signaling negative. Interestingly, if you culture the cells in inhibitor of notch, then you accelerate the process of differentiation with these cells. So again, it seems that notch is rather inhibitory and for cardiogenesis and more promote the proliferation of undifferentiated cells. Now in the heart, in the adult heart, where you find notch signaling cells as detected here by an antibody directed against the intracellular domain of notch, in cardiomyocyte obviously, but also in non-myocyte cells. So both types of cells signal through notch. The ligand is found on the fiber mostly, on the cardiomyocyte, and they have maybe more expression in area when you find these NKX 2.5 positive non-myocyte cells that represent the cardiac precursor cells. Now the ligand is upregulated with stress, with pressure overload. There's an increased ligand expression. Suggesting that it is important in normal adaptation of the heart to activate notch in adjacent cells. So basically, there's different situation here because the ligand on the surface of the cardiomyocyte can signal to the adjacent cardiomyocyte, but also to the surrounding cells. And we interest obviously on the behavior of fibroblasts because these cells lead to fibrosis at the end in a normal situation. But the ligand could also recruit cardiac precursor cells. And the question we ask is whether by changing the concentration of this ligand on the surface of cardiomyocyte, we could maybe tip the balance towards something more regenerative by favoring the recruitment of cardiac precursor cells. And I will briefly now describe a transgenic model in which we have tried to assess this. Now, it's very simple. We just use the alpha-MHC promoter to overexpress JAG1 on the surface of cardiomyocyte. So here you have the quantitation of the expression. So it works. On immunostaining, you see that all cardiomyocytes express the ligand. And at the end, you have increased signaling in transgenic animal as compared to wild type, both in non-myocyte cell and in cardiomyocyte. And this is a quantification. Now, what is the phenotype that develop in these animals? Well, basically, what you have is an increased cellularity in the ventricle wall. So more cardiomyocyte. And this is likely due to an increased proliferation in the first week after birth in the cardiomyocyte population. So you have a sustained proliferation after birth in these animals. This is a phosphor H3 staining in cardiomyocyte. Now we prove the point by doing a BRDU pulse chase experiment when we inject BRDU in neonates and apply a 30-day chase and observe these young mice. And what you observe is that, yes, there is indeed an increased production of cardiomyocyte in the transgenic animals, but also there's more non-myocyte cell that is produced, and these cells are majoritarily NKX 2.5 positive cells, suggesting that you have also an increased production of precursors in these animals. Now, this is the key experiment. What is what's happening in the adult animals when you stress the heart? So we use the transaortic constriction model here, and same time we administered BRDU and observe what are the cells that proliferate in this under this condition. Now there is obviously proliferating cells in both genotypes. However, there's a major difference. In wild types, what you have is a big increase in myofibroblast production. And this is observed here also by immunostaining. And this doesn't occur in the transgenic animals. And if you, on the contrary, I'm sorry for the panel here, but this is an important finding. there's more production of non-myocyte cells expressing NKX2.5, so more precursor production. Now we found also, we used SCA1 staining for detecting precursor cells, and what we found is that in transgenic animals, you have more SCA1 positive cells that are produced. You have also more SCA1 positive cell per cluster of positive cells, and we have more cluster of SCA1 positive cells in these animals. Now, importantly, it seems that the SCA1 positive cell receive notch signals, but these cells are essentially NKX2.5 negative. Now, in order to and if you look at NKX2.5 cells, they are SCA1 negative. So it seems that you have a conversion between SCA1 positive cells receiving a notch signal and precursor cells that express NKX2.5, consistent with the finding we have found in stem cells in vitro. Now we quantify these cells carefully in the heart of wild type and transgenic animals, and maybe we can concentrate on this particular differences here. Again, the notch IC negative cells, NKX2.5 precursor cells, are largely increased in the transgenic animals as compared to the wild type. And this is not dependent on pressure overload. Now, what's happening in terms of adaptation of the heart? But basically, these animals demonstrate very much attenuated hypertrophic response to stress. This is what happened in the wild type situation. The hypertrophy is evident. You have a shift in the cardiomyocyte size distribution here. And this is not observed in transgenic animals. Now you can quantify also the size of the cardiomyocyte. You see that there's very much different. They are very much different in the transgenic, smaller, and at the end, left ventricular mass is not increased after stress in the transgenic animal. Now, the other component of the response, fibrosis, is also decreased, and you have here staining for TGF-beta2 and CTGF. In wild types, it's evident that you have an upregulation of these two factors on the stress, and this is not observed again in transgenic animals. And this is the quantification of the expression. Finally, there's also less fibrosis that develop in these animals. This is the wild type, this is the transgenic, and if you quantify fibrosis, you see that you have much less. So the message here is that this animal has a better capacity to deal with pressure, and this is most probably due to the increased capacity to produce cardiac precursor cells. Now, obviously, there's many steps in this process in which not could be operational. Our findings seems to indicate that the mesenchymal stromal population is the prime target of notch signaling in this situation. If we take off notch 1 of the equation by deleting notch 1 on cardiomyocyte, basically the phenotype is maintained. So it seems that the signal, primary signal here is directed to the mesenchymal population. Factors are obviously involved. Maybe they are downregulated in the situation, and we have a shift between a more fibrotic type of repair and to a more regenerative repair mechanism. Okay, so now I'm going to switch gear and talk about a little bit our new attempts to identify new pathway that could be important in cardiac regeneration, cardiac adaptation. And as like many other, we took advantage of the zebrafish model. So we have a presentation already about this model, but basically if you remove about 20% of the ventricles on these animals, within a few weeks you have a complete regeneration of the myocardium. So it makes this model organism an ideal organism to test or to identify pathways that could be important in inducing regeneration. So there's it's a complicated process, actually, regeneration in this organism. It implicates different cell type and different pathways, different factors, which culminate at the end in the proliferation of the differentiated cardiomyocytes, as we've heard already. But what is interesting is that all these pathways actually are also used in part in the mouse heart under stress. So there's common pathway, but they are differentially utilized. And the question is why they are differently utilized and how can we identify the regulator of this pathway. Now, this pathway under control of the gene regulatory network, cardiac gene regulatory network, and this is also governed by a battery of transcription factor, but also by different molecules, and specifically non-coding RNA. So I've indicated here several, but I'm only going to talk about today about microRNA. There's probably more and more evidence for the importance of long non-coding RNA or enhancer RNA in this situation. But today I will focus my presentation on this experimental approach to try to identify microRNA that can regulate differentially common pathway in the mouse and the fish. So it's a basic comparison of these two organisms and in the heart to identify common pathway between the mouse and the fish after injury in the heart and during the fibrotic repair on the one hand and regenerative repair on the other hand. So what we did is that we took the myocardial function model, isolate RNA from the border zone, and apply a microarray analysis and a small RNA sequencing to detect microRNA. And we compared this the data with suits obtained in the regenerative tissue of the fish after 2 weeks, and apply again microRNA and small RNA-seq to identify microRNA. So these are the first data. So this is just to show that the system works. Obviously, we have the expected remodeling, decreased function in these animals. We took 6 sham-operated animals versus 6 MI mice, and you have the expected upregulation of the fetal gene program. We observed modulated, we detect modulated gene, but 6,000 genes, and modulated pathway as expected. Now, in terms of microRNA, we have identified 94 differentially modulate microRNA in this situation. Now, we detect the obvious one that has been already described in the literature. One important point is that if you do clustering analysis, there's a very good homogeneity between these different samples, suggesting that you have really a clear response of the microRNA transcriptome under this situation. Another interesting point is that the most frequent RNA, the RNA that are easily detected, high expressor, are not the most frequent one, because there's obviously a lot of modulate microRNA that are expressed at low levels. Maybe they are expressed in specific subtypes in the heart. So that will be a challenge then to find which subtype they are expressed in. Now the fish, so we took the regenerative tissue from 50 fish and create pools. I mean, we used 4 pools of 50 fish, each sham operated or after resection. There's about 1,000 genes that are differentially modulated in this situation. Maybe less genes because we're using pools, so maybe the detection limit is a little bit blunted. But we have genes that have been shown to be important in regeneration that's been described in the past. Now we found 27 differentially modulated microRNA in this situation, and again, clustering analysis showed the homogeneity of the samples. So how we can use this information now? So this is the list of the 8. Obviously, you find some that have been described in the past, but there's new ones. And all of these microRNA have been implicated in different cell processes. Now let me show you a few things. Now this is subnetworks that we have extracted from this list. Now the list of the gene is not very interesting for now. But what is important is to show that you have obviously microRNA that are higher in the fish than in the mouse. But more importantly, and some that are lower. More importantly, what you see is that you have this positive and negative correlation. So it means that you can have targets of MIR that are upregulated when the MIR is upregulated. And this is an interesting situation because there's maybe explanation for that. Now, there's also MIR that are apparently modulated the same way. Interestingly, if you look at the subnetwork, they create a subnetwork in themselves, and they have cooperative subnetwork that can be extracted from this information. Now, all the expected targets are there, but in addition, you have common target between these MIR, 2 by 2, or even if you consider all of them together. And the one that are predicted here, for instance, GLI2, a component of the sonic H pathway, or fibrin 5, which is implicated in the stabilization of the matrix. Now we have confirmed this in samples. So again, in the mouse, these microRNA are downregulated. The expected target are upregulated. And this is the contrary in the fish. And we confirmed the modulation, the direct implication of the MIR in the modulation of this target by cloning the 3' UTR of each of these genes downstream the luciferase gene and do a luciferase assay. And you can see that if you overexpress the MIR, you have a downregulation of the expression of this gene, of both of them. Now it's interesting because we haven't pursued this further, but it's already been described that GLI2 can associate it to MEF2C to control differentiation maturation in the heart. And fibrin 5 is obviously have been shown also to be important in the normal adaptation of the heart. So you can speculate about the effect of this, but it's obviously a different outcome that will result from the modulation of this gene in the fish and the mouse. Now, one interesting example is MIR26A. MIR26A is not changed in the mouse. So if you would just do a mouse screen, you wouldn't detect it. But it is downregulated significantly the fish. Now, this microRNA has not been described in the heart, but has been described in cancer field, and it is actually a modulator of the cell cycle. It is actually embedded in a phosphatase that dephosphorylates RB. And together, because it targets important regulator of the cell cycle, then it can modulate the whole cell cycle and maybe contribute to the inhibition of the G1 to S transition. Now, the second important target of MIR26A that has been described is ICGEB. And ICGEB is a member of the polycomb complex PRC2. Now, this protein has been recently shown by 2 groups to be important in the heart during development, but also in postnatal cardiac homeostasis. And it's important, gene repression that is established during development is important for adult tissue specificity. So hundreds of targets has been identified for ICGEB in this situation. The non-cardiac gene, obviously, but also cell cycle regulators, inhibitor of the cell cycle in this case, and also the fetal gene program. So, for instance, these proteins are target gene of ICGEB in differentiating cardiac progenitor. So, as with the first preliminary experiment, we just looked at the expression of MIR26A after birth, in the heart after birth, and we observe very much increase in this expression at the time while the cardiomyocyte exit from the cell cycle. If you take two or three specific points here, that you see that you have, expression of this MIR during the first week or so after birth, and that correlates with the expected target gene downregulation in this situation. Now, what we have done as preliminary experiment also, we've used an inhibitor of the MIR and administered that to a mouse, an adult mouse, after and looked at the response to myocardial infarction. Now, the MIR is very, 2 weeks, we sacrifice the animal in this case. So after 2 weeks, what you have is a clear inhibition of miR26A and a clear upregulation of ICGEB. And of other target of miR26, that is the regulator of the cell cycle that I have shown before. Now, if you look at the ICGEB expression by immunostaining, then what you see that it's not very much expressed in animal injected with a scramble inhibitor. If anything, it is expressed in dividing non-myocyte cells. But when you inject the inhibitor, you have very much upregulation of this protein, particular protein in cardiomyocyte. Now, finally, this is my last data slide. You also observe the expected downregulation of the fetal gene program in this situation that you would expect if ICGEB is indeed involved. So obviously, I'm not saying that this is the only target, by no means, but now we have to obviously to find a way of identifying the ICGEB targets in these two situations to identify important therapeutic targets. So this is a summary slide. So we think that miR26 modulate ICGEB, but also clear modulator of the cell cycle, and by such modulate hypertrophic fibrosis and proliferation in the myocyte population. Okay, so I think we have established a pipeline for identifying important microRNA in the heart, and hopefully in the future we will detect more important of this microRNA that could be used as tool for inducing regeneration in the heart of the mouse. This is my analytical side, so people that did the work, Stéphane Agripin, Mohamed Nemir, Samir Hunzain, and Gaël Boisset did the fish experiment, and we are very grateful to Mark Ibbotson for the bioinformatical analysis. I mean, these guys spend weeks trying to analyze this data, and that are summarized in just one slide. So thank you for your attention.